Well, welcome everyone. Thank you so much for joining us on our March edition of the CellCAN webinar series. This current stop takes us to Edmonton, Alberta, to hear more about the journey to the clinic with the Alberta Cell Therapy Manufacturing with our excellent speaker, Gayle Piat. As people are joining on, everyone should be joining muted, and I will make a brief mention quickly right off the bat that if you hover over the window in the middle, especially the top right, you'll see a chat function. At any point in time, please don't hesitate to interact with us. Send us any questions that you may have, and I'll certainly do my best to make sure that at least by the end, during our discussion period, that any of your questions or discussion points are brought up and addressed. So before we get started too much, I'll make a few mentions of CellCAN for those of you who are not familiar with our Regenerative Medicine and Cell Therapy Network. We were established in 2014 by the Networks of Centers of Excellence as a not-for-profit focused on knowledge mobilization around the practices and of good manufacturing practices, involved in cell and gene therapy manufacturing. Our vision is that cell and gene therapy manufacturing in Canada must operate under a common seal of quality to increase capacity and rapidly and effectively migrate innovative treatment concepts into standard clinical practice. Our mission is to improve the quality, safety, and feasibility of cell and gene therapy in Canada through focusing on optimal manufacturing practices. We have 4 business priorities: to build a strong Canadian network, which includes breaking down the silos between cell and gene therapy manufacturing facilities and cell and gene therapy stakeholders, certainly in Canada and globally. To advance regulatory standards, we work closely with the BRDD at Health Canada to focus in on points that require immediate attention. Generate effective outreach through mechanisms such as this webinar series and our strategic forum, which we do have coming up in 2022 in March, so a year from now in Ottawa, when we are all immunized and pandemic restrictions we hopefully will see loosened. And as well, we want to position Canada as a world leader. We're really pleased to report that our Advanced Training Institute has launched. So if you haven't seen or heard already, please don't hesitate to go to cellcan.com or atmptraining.com, where you can see a list of the various offers that we have online. We have over 20 courses available right now. The list is growing and actively being updated, so look for new announcements. You may have seen our newsletter, which came out yesterday, outlining some of this information. We have 2 learning paths in particular that we've assembled the courses into. The first is an essentials of CGT GMP manufacturing in under 60 minutes, more of an executive level snapshot of what GMP entails and how it impacts our field of cell and gene therapy manufacturing, and as well the highly acclaimed hands-on GMP workshop we put in virtual format. It's an interactive computer-assisted learning based course focused around adult learning principles. It's highly interactive with different assignments and documentation around it, which give a certification at the end, and it's designed to be completed in under 20 hours. The interactive modules, as I mentioned, help guide you through the learning process, give you an update on the type of progress you've made so far, and our professionalized content will help elevate the level of interactivity with the program. The assessments are knowledge-based, so it's intent on knowledge uptake and giving you the foundational knowledge and basics around different types of manufacturing, different types of equipment of manufacturing, and of course the foundational principles around GMP, such as quality management systems, validation, and quality control, as well as product release. We're pleased to report that our training program has been recognized by Engine's AMP Up program. For those of you not familiar with it, this is the Next Generation Manufacturing Canada, a supercluster from the federal government. They have a program focused on training, which we've been an approved training partner, which If you're a manufacturer in Canada, you're eligible for 50% of the training costs covered by Engine. It's free to become a member, and please visit engine.ca/upskilling for further details. Now, without further ado, again, for those of you who just recently joined on, I will mention that if you want to pose any questions or include any comments, the chat is located in the top right of your window. And I'll go ahead and introduce our speaker. So Gayle Piat has over 30 years experience working in the life sciences field. She has a background in medical laboratory technology and spent 12 years specializing in certification under good laboratory practices. In 2003, she earned the designation of Registered Quality Assurance Professional in GLP through the Society of Quality Assurance while working at Alberta Research Council as the Quality Assurance Manager at a GLP toxicology facility. In 2011, Gayle joined the University of Alberta as project manager for the construction and initialization of a GMP cell manufacturing facility, the ACTM. Gayle is currently the manager of the ACTM and the person responsible for translating projects under good manufacturing practice for clinical production. In 2016, she further received the Regulatory Affairs certification through the Regulatory Affairs Professional Society. She's a very active member of our CellCAN network. We're very pleased to have her today to present her progress in Alberta. Gayle, I'll turn it over to you to take over.
Okay, thanks very much for the introduction, Craig, and thanks everyone for attending. Share my screen here. This is always becomes the hardest part of presenting, I think.
The new age of presenting. Perfect. Now we see your slide deck and you're in presenter view. Take it away.
Okay. Great. Okay, so I'm going to introduce you to our GMP facility in Edmonton, Alberta Cell Therapy Manufacturing. And so our facility is located on the campus in Edmonton. And you can see it's very closely, we're in this building right here. Craig, can you see my arrow on the screen?
Yes, absolutely.
So we're in this building here, the Li Ka-shing building, and you can see that we're situated very close to the University Hospital too, which is great for collaborating on clinical trials. So we're in the 7th floor. Our facility's on the 7th floor. When this building was first constructed, the Faculty of Medicine had a GMP facility in mind, but they didn't have the funding at that time. And so they left the 7th floor blank and the 8th floor, which is an interstitial space, with the hope of having a GMP facility there someday. So in, I think about 2008, Dr. Greg Corbett wrote a CFI application to build a GMP facility, and he was successful in receiving that funding in 2009, and then received supporting funding from the Alberta government in total of $26.2 million to build the facility. So then there was a few years of design considerations and getting construction team hired, etc. So construction was initiated in 2012. I joined the university in 2011. So construction took a while, as it's a complicated facility to build and lots of checks and balances that have to be done and commissioning. And so we had facility takeover in 2015, and we began to hire our staff. 2016, we were continuing to do facility setup. Anyone that's been involved in setting up a GMP facility knows for sure it takes a long time to do equipment validation and qualifying the facility itself, writing SOPs, and developing the QA program. So in 2017, we had our first project initiated, and then after that, we continued on further with more projects. So just to give you a little snapshot of history, this was what the 7th floor looked like before we began construction. And this was during construction, and this is what one of the clean rooms looks like now. On the 8th floor above, as I mentioned, it was an interstitial space, so that's where all the mechanical space is that feeds into the clean rooms. So this was before, and this is what it looks like now. It's like a city up there with all of the ducting and whatnot that is required for the operation of a clean room facility. And on the right here is one of 4 air handling units that we have. So a little overview of our facility. It's 10,000 square feet, so it's about half of the 7th floor of the building. We have 6 grade B clean rooms. We have a QC lab, a process development lab. It was designed with so that we can do unidirectional flow to prevent contamination and cross-contamination, since it was designed for cell therapy products. We have material pass-throughs all throughout the controlled areas. And it was the facility was fully qualified by an external contractor. So just a little virtual tour to our building in Edmonton. Remember these things that used to fly in the air? So we're mainly sunny in Edmonton right now, and I think it's a high of 15, so spring is on the way, and here we go into our facility. So this floor plan, the white part of this floor plan is our facility. The gray is another research group. So when you come up to our facility, you can see the entrance to our facility just where the arrow is pointing. So this is the entrance, card-locked entrance in, and the blue area is our office area, and the yellow is our archive room where we have fireproof cabinets for all of our records. So just inside the facility, we have a material management area. So that's where all of our materials are received when they come into the facility for production, and they're quarantined and released. We've got fridges, freezers, and ultra-lows in that area. Then just down the hallway, we have our QC lab. So it's well equipped with a lot of different equipment, but we're kind of bringing them on board for GMP because it takes a long time on a project by project basis based on requirements. So currently we've got our Beckman Cytoflex for cell characterization. That's kind of our workhorse. So we've got that up at GMP level. And then we've got a Bactylert for sterility, and Charles Rivers PTS system for endotoxin. And then, of course, cell counting and viability is a constant QC test that's done as well. Then next to the QC lab, we have a material preparation room. So we've got back in the corner here, this is one of 3 GMP autoclaves that we have. We've got a plasma sterilization system here, and then just an area with, dishwasher and a still for water for our incubators, etc. So that's all uncontrolled space. So then we move down the hallway into the controlled space, so the clean room area of our facility. So the color coding is designating which areas are served by the 4 different air handling units in the facility. So just to show you the flow, so the arrows are showing how you get into the facility. So there's a gowning room, intermediate entry. Then there's airlocks that you can go airlock in into the clean rooms, and then airlock out when you leave to an exit room to degown. And each of the areas have their own gowning room and exit room. So just to show the flow so that we're controlling personnel, materials, product waste all throughout the facility. So just a little snapshot of what the gowning rooms look like. So there's 3 different gowning rooms depending on which clean rooms you're entering in. So when we designed the facility, we were able to ask the contractors to make put a square. So that's where you do the swing over bench to put on clean booties and then enter into the intermediate entry. And as you can see, there's pass-throughs here to pass materials into the clean room. these are the intermediate entry rooms, just past the gowning room. So these are equipped with freezers, fridges, and ultra-lows so that we can move material that has been released for manufacturing into this area so people can pick up materials as they're moving into the clean room. So this in the photo here, this would be a gowning room. So you would go through the door, retrieve your materials here. This pass through at the top here, then goes directly into the clean room. And this is the airlock in that you would go into, and then the doorway into the clean room. So then after you're through airlock in, you're inside the actual clean room. So our clean rooms are all about the same size, and they're equipped with biological safety cabinets, 4 to 6 incubators, tri-gas incubators, and then centrifuges and microscopes. And then we have specialized automated cell therapy equipment that can be moved into the rooms as well on a project-by-project basis. All the five of the rooms exit into this exit room here. We've got a 6th clean room over here that has its own exit room. So in this room, there's a waste pass-through back here in the corner. So that leads into a waste autoclave room where we deal with all of our waste. So we've got a separate flow for waste versus the product that would go out through the degown area. And then we also have our controlled rate freezer in this area and our storage, liquid nitrogen storage. And our dewars for the source liquid nitrogen are on the other side of the wall. So the liquid nitrogen is piped into the clean rooms, but we don't have to manage having cleaning a dewar to bring it in and out of the facility. It's just piped through the wall. We have lots of equipment. So we've got 30 tri-gas incubators, we've got 18 fridges, 9 freezers, 8 ultra-lows. We've got a 4 and 6 foot VSC in each clean room. We've got benchtop centrifuges and ultra centrifuge, a high-speed micro. We've got, as I mentioned, 3 GMP autoclaves and a waste autoclave, plasma sterilizer, pass-through dishwasher, and phase contrast microscopes in each room. And then some of our more specialized equipment, we've got a Prodigy, a Clinimax Plus, we've got bioreactors, we've got a Wave, we've got a Lovo cell washer and a Lutra. We've got an Active Pure and Active Flux for protein purification. And we've got 2 controlled rate freezers, and then, of course, liquid nitrogen storage freezers. We've got a routine cleaning and maintenance program, an environmental monitoring program, of course, like every clean room has. And we've got a bonus cleaner, our robot, which we can use for changeover if required between projects. So we shut off our air handlers and tape all the doors shut, and then we put the robot in the clean room, and it spews out vaporous hydrogen peroxide out of its head here and kills everything in the room. So we can even open up all of the incubator doors and fridges, and the vaporous hydrogen peroxide gets into all the nooks and crannies that can be difficult to clean manually. We've got a facility monitoring system that's computerized, so it's monitoring all of our equipment and our rooms 24/7. So you can see the map of our facility on the right here. So if anything goes outside of the specifications that we've set, we get text messages on our cell phones, and the map will go yellow or red, depending on what the value is. And then you can click on the map and zoom in to see exactly what piece of equipment or what parameter is out of spec. So when we went through starting up our facility, we looked at each piece of equipment, and so we want to make sure that we have good control over our equipment. So I'm using incubators as an example here, but so incubators, of course, are a workhorse for a lot of our projects. So we looked at building in redundancy. So we have 4 to 6 incubators in each clean room. We've got a gas manifold up on the 8th floor that our gases are piped into our clean rooms through turrets. And we've got redundant cylinders, so it automatically switches from one cylinder to the next. And then when it switches from one side to the next, we get an alarm on the 7th floor, so we know it's time to go and replace the cylinders. So we know that we're never going to run out of gases in these precious incubators. The incubators are all on emergency power, and they're monitored for temperature, humidity, and CO2, and alarmed 24/7, as I mentioned. And then we fully validate them. So in this case, we were using hyperflasks. So we've got a K validation system with these wireless data loggers. So we program them and put them in the incubator and then monitor the temperature by mapping the temperature in various levels and corners of the incubator to make sure that the temperature is consistent. So our team at ACTM, so we've got independent quality assurance who reports directly up to the VP of Research. We're under the Faculty of Medicine and Dentistry. And Dr. Greg Corbett is our scientific director. I'm the manager, and then we have a team of manufacturing staff and QC staff. Because we're part of the university, we have a lot of support groups that look after things like contracts and legal and managing purchasing and procurement, HR, financial services, safety, facilities and operations looks after all of our air handlers, and then we've got IT department. So just a little bit about our staff too. We were fortunate that we've got a couple members who have a lot of years of experience in industry, come from biotech, including our QA manager. And so that's been really helpful so that they were able to mentor some of the other staff that we brought on board in the beginning and set up the quality assurance program. And we've got staff that are very talented in cell culture and cell banking and QC assays and assay development and qualification and equipment validation and environmental monitoring. So it's a great team. We haven't had high turnover, so a lot of them have been there from the very beginning, and very strong team and very, very dedicated team. So I just want to throw out a huge thank you to the team at ACTM for all of their dedication and hard work. So our training program, so when we bring new people on board, they receive GMP training, and hopefully they've had some past GMP training, and that's where CellCAN's training program that they've developed would be really key for people to have that training, so you have a good base before coming to join. Or having the training after always works too. So there's 4 types of SOP training that we have incorporated for our SOPs at ACTM. So it might be read and understand if it's more of an administrative type of SOP. Might be discussion or demonstration. It might be hands-on. And for some of our SOPs, we actually have competency where you actually have to pass certain criteria before you would be allowed to do that procedure. We have retraining requirements in our SOPs. So when they're revised, we decide in advance what type of retraining is required, if it's just read and understand, if it's just a small change, or if you have to redo competency because there have been major changes. So an example would be gowning qualification, where first the trainer demonstrates, then the trainer observes the trainee doing it, then the trainee has some time for practicing, and then the trainee goes through a test and they gown, and then they're sampled with contact plates and they're incubated to make sure that there isn't any growth, and then they would be deemed competent to go work in the clean room. And then there's a requirement for the retesting on an annual basis. So just quickly, a little bit about our QA program. We've got more than 90 SOPs and growing all the time. Internal audit program, oversee the program of quarantine and releases releasing materials used in production, and oversight of QC testing and actually releasing the final product, issuing all of the GMP documentation, looking after training files, oversight of the validation of equipment, the calibration and maintenance program, vendor approvals, investigations, and CAPAs, which stands for Corrective and Preventative Action, incorporation of all of the regulatory and guidance documents. So full QA program that definitely took a while to build. Now, just a little intermission, a little fun fact. I actually live about an hour out of Edmonton, so I commute into Edmonton to work. So the town I live in is Vegreville, strong Ukrainian town. And so since Easter's coming up, I thought I'd share a picture of our Easter egg in Vegreville. It's the second largest Easter egg. Pisanka used to be the largest, but now Ukraine has the largest one. But it was built in 1975 to commemorate the centennial of the RCMP. Now back to ACTM. So working with ACTM, we follow a standard pathway that any contract manufacturing facility would follow. So first, before we can start talking about details, we have a confidentiality agreement signed, and then we look at the project scope and we issue a quote, and then the client can come and audit us, and then we put out a quality agreement with the client and a service contract. Then there's tech transfers, so we get protocols that have been developed already by the client and any assays, and we work on them at ACTM and incorporate them into our documentation system, and then we qualify any assays that are required. Then we would develop material specifications so we can release all the materials that are used for the process, and then approve any vendors, develop the batch records and SOPs, do some qualification runs or engineering runs, and then go into GMP production. Of course, security and confidentiality is very important in a GMP facility that's doing contract research. So we've got card access all throughout the facility. We use project codes on all of our documentation and labeling, so the client name is not on documents that are around that people might see. And then all staff maintain very strict client confidentiality, of course. So a little bit about a QA agreement between the client and ACTM. So it's an agreement so that we can really delineate the responsibility that each party is taking for different GMP aspects. And really it's so that nothing is falling through the cracks and there isn't any assumption that one of the parties thought the other party was responsible for it. And so it would include also product specifications and key contacts for each party. And if there's any testing that has to be done that's subcontracted, then just really highlighting who's doing the contracting and managing that testing. So a little bit about our manufacturing capabilities. So we do manufacture cell therapy products for clinical trials. We have done vaccine products for clinical trials, cell banking. We're setting up to do aseptic fill finish, and I'm going to talk about that in a little bit, and then QC testing. So for cell therapy product manufacturing, for the cell culture, we have several different options depending on the project. So we've got, as I mentioned, the WAVE. We've got a quantum bioreactor system. We've got Miltonese Prodigy. We've got a bioreactor system from Eppendorf. And then, of course, we've got incubators so that we can do flasks and bags and standard incubation. Then we've, for cell separation and cell washing, we've got the choice of the Lovo, the Elutra, the Clinimax, or standard centrifugation. Then cryopreservation, we've got controlled rate freezers and quite a few different dewars for vaporous liquid nitrogen storage. So I wanted to just highlight here again that we're moving into future fill finish capacity. So this suite that I've highlighted here is on its own air handling system, and it has its own gowning room, its own intermediate entry airlock into our 6th clean room, airlock out, exit, degown. So it's the perfect suite, I guess, to have aseptic fill finish because we don't have to worry about any interactions with the other projects that are going on in the facility. So right now, we do manual fill finish for our cell therapy products in the same room that we would be doing the manufacturing. So if we were making a master cell bank, we would clear out everything and do the fill of the vials right in the BSC in the Class B or Grade B clean room. But this separate suite would allow us to manufacture in those clean rooms and then move into the fill finish suite. Or even if we weren't doing the manufacturing, to receive bulk product from another manufacturing facility and then fill that product in our facility on a contract basis. So we currently have a semi-automated system. It's from Aseptic Technologies out of Belgium, and it's called the Crystal M1 filling station. And these are just an example of the different vial sizes that the system can use. And so they say that you can do about 180 vials an hour, so you could do up to 1,500 vials per shift. And we are potentially looking at applying for funding for a more robotic system as well. So hopefully that will come as a phase 2. So I'm not sure if this video is going to show up very well. It wasn't streaming very well, but I just wanted to quickly show a video of what the system that we have now looks like. So it uses a peristaltic pump. It's got a completely disposable tubing system with the needle attached already that you would install in this system. And then you set up the volumes that are going to be dispensed. So you choose your volume and vial based on the project. Then this is what the semi-automated part. You have to manually put the vial on the system, pull down the lever, it dispenses into the vial. And then right next to it, there's a laser system. So you put the vial in this box, press a button, a laser shoots at the top of the septum and seals the hole that the needle made in the top of the septum. And then you manually would put the cap on. So the robotic system that we're looking at, you wouldn't need all of this operator intervention. You would have a robotic arm doing all of these steps. So we have a small process development lab, and we've got Eppendorf's DASGEEP system so that we can have 4 different bioreactors running at the same time and alter the different operating conditions, whether it's the stir rate or the different medias or the all different types of parameters so that you can optimize your process. And then once you pick the process, then you can move it up into the bigger bioreactor system in the clean room facility. And then we can just do other even non-GMP cell banks we've done in here, or if we want to do an engineering run and we have the equipment in this room, we could do a preliminary run before we go into the clean room. So we've done cell banking for various different clients, GMP and non-GMP cell banks. So we've done SF9 insect cells and mammalian cells, Helo and Vero. And so all of this cell banking we do with using a batch record and calibrated equipment. So even the equipment that's down in our process development lab, we use similar SOPs and maintenance schedules, et cetera. So still keeping good documentation practices, et cetera. So as I mentioned previously, we have high capacity. We have lots of incubators and they're long-term storage as well. We've even stored some things for clients, even if we haven't manufactured them, just for long-term storage in ultra lows or liquid nitrogen freezers. So our QC capabilities, as I mentioned previously, the ones in blue are the ones that we have set up for GMP, but we do have various other types of equipment available that we could set up for GMP if a product requested it. For instance, we've got a CDX bioanalyzer, and so that's used to test the media in bioreactors so that you can monitor glucose and lactate, for instance, so you can optimize and know when is the best time to change out your media. This is just an example of the assay qualifications that we're able to do. So this was for annexin and PI viability. So first we write up a protocol and it goes through a tech review, and then there's approval by management and the client and QA. And then the protocol will explain what we're doing. So in this case, we did precision. So we tested 3 different cryovials on 3 different days. We looked for intermediate precision by having 3 cryovials analyzed by 2 different analysts. We looked at sample stability, repeatability, robustness, specificity, and then we write a report. And then all of the raw data and report are reviewed by a technical reviewer, by management and QA. And then all the raw data and report is scanned and sent to the client for review. And then the report is reviewed. And then after all of that is done, then you can take all the information that you learned and incorporate it into the SOP. So some of our current projects that we're working on, we've started a CAR T cell phase 1 trial. So our first patient is going to be dosed at the end of this month. We're doing an engineering run for a cell therapy product for a potential phase 2 trial. We're doing another engineering run for a phase 1 clinical trial. We've worked on thymic Tregs for a potential phase 1, and we've manufactured a COVID vaccine. You might have seen it in the news with Antos Pharmaceuticals for a phase 1, and we've done cell banking for various U of A researchers, and currently there's a hepatitis C vaccine being manufactured with Dr. Michael Houghton's group at the U of A for a phase 1 as well. So that brings us to the end of the presentation. So now I'd like to open it up to any questions you may have.
That was fantastic. Thank you, Gayle. It's a tremendous amount of work, and a great amount of congratulations are in order. I know it's quite the process to initialize a small facility, but that's got quite a spread of different projects underway, and a lot of work has been put into it. So congrats. Now I'll just remind everyone that there is the chat function on the right. Please don't hesitate to populate your questions if you want to interact. It'd be great to engage with Gayle and discuss the various aspects of her presentation that she covered. And maybe I'll start it off. Actually, before I do, I'll mention I'll put up a slide in a moment here, a brief survey. If everyone can take a moment to contribute to the survey at the end, it helps us to really focus in on the content. But I'm obviously, we talked about CellCAN. Gayle, you're a very active member of our education committee who helps form our curriculum to train HQP and GMP manufacturing. And you mentioned that you were able to bring on some QA personnel, especially from the biotech sector with relevant experience. But can you maybe just make a few comments about what that experience was like when you're initializing your facility, you're identifying the roles that you require, you start searching for candidates, and where do you find them, and how difficult is that process?
Right. Yeah. I think staffing for GMP facilities probably worldwide now is a challenge as people are gearing up and getting more and more projects. Like right now, our clean rooms are full. And so we are needing more staff because we're growing, and we're going to be hiring 3 more people for our fill finish suite to start that up. And so there'll be another QA person and two operators. And you're quite right. Where do you find the people that have the previous experience? There isn't a QA school until maybe now that CellCAN is helping to set one up, or already set it up. But people learn mainly by experience traditionally. So you're brought in and you're mentored. So you when you come into our facility, as I mentioned, you start with a base GMP training program, then you're trained on our SOPs, and then you're really, teamed up with another staff member, and they have to, just slow process of training up on each SOP and demonstrating it and practicing it and etc. What would be fantastic is if people were already trained in aseptic technique, and that they had demonstrated through a media fill. So when you take media and then you go through a process, and it's media that, like a tryptic soy broth, so that it's meant to grow bacteria, and you simulate and pipette and incubate and then transfer things from a tube to a centrifuge, and then take off the supernate, and you're simulating all these steps, and then you incubate that broth and show that you didn't contaminate it. That is how we prove that people have good aseptic technique, because people can think they do, but in GMP, you have to prove everything. Like, we don't make any assumptions. So if people have gone through training and they are able to apply for a position at our facility, and they can say, I've already demonstrated that, I've already gone through that test and I passed, then we don't have to just guess, gee, I wonder if they're going to be good at aseptic technique based on an interview or based on references, right? So that's what I think is key, is you can read the regulations and you can read it in theory, like you can just understand it, but it's actually putting those regulations to use in practice is what is really key to having good training and good skills, and be able to hit the ground running when you come in to work in our facility. So that's for more of the hands-on thing, and cell counts and all that sort of thing. That's the production. But then there's QC assays. That's another role. And there's so many different roles in a GMP facility. People tend to just focus on the manufacturing part, but QC, there's people that specialize in developing an assay, in qualifying the assay, and then in actual running the assays in a GMP compliant way. Then there's quality assurance staff, and that's a completely different set of skills. And they have crazy attention to detail. They understand the GMP requirements at a depth that's required to know when you bring in a new project, are the materials going to be compliant? Are the assays appropriate? Looking at the batch records and the process itself, and deciding which parts are going to need to be brought up to a higher level of GMP. And there's so much preparation that's involved in getting ready for a GMP run, then actually doing the run, and then reviewing all the documentation after the run to release the batch. and that's QA's role. So their role is to look after all of the GMP oversight for just the day-to-day running of the GMP facility to make sure it's ready for a project, and then to look after the GMP compliance for the project itself.
It's very much an ongoing assessment of the risk, a calculation of the risk, and assigning a value of the risk, would you say?
Yes, a lot of risk assessment is what's going on in that, and working with the client too, to collectively decide where the risks are, as in a partnership, really. And then there's equipment validation, there's environmental monitoring. So for someone to say that they've had experience in GMP in another facility, it's always, okay, what type of experience? And if that experience can then be translatable over to the type of position that we're asking for. Then, so those are all GMP type. Then there's technical experience. Have you done cell culture before? Have you done QA/QC assays before? And then there's very important skills that are just soft skills. Not everybody is suited to work in a GMP facility. Some people do not like it at all. Some people love research and development because of the creativity, because of just the ability to go off onto other avenues because you've learned something and there's something interesting and now I can go to this. Very rigid, but it's also very rewarding because you are taking that R&D that somebody has spent sometimes their whole career developing, and you're taking it into that next step to bring it into the clinic. So
absolutely.
I hope I've answered your question. I rambled on a little bit long.
No, you've hit on all the right points. And actually, it's interesting when you put the focus on quality. There's been quite a bit of discussion I've had recently with working with quality team members, so quality assurance, to help give them more practical process-related experience. Because as you say, it is a very different job role. Many people come from the sector, they come from quality. They don't always have the same caliber, I would say, of on the floor type of processing experience. So it's an interesting point of view. We've had a couple questions come in that maybe we'll shift the focus. And Ken has asked if he's curious to know more about the fill finish. Is it potentially the VanRx system?
You know what? Would love to have the VanRx system. We can't get it into our facility due to size.
Yeah.
We can't get it through the doors. We can't get it up our freight elevator. It would have to come through the wall. So we've tried a couple times to look at ways that we could get it into our facility, and just can't come up with a solution. So yeah, regrettably, it will not be.
Right.
I would have loved to have, support a Canadian company right next door in Vancouver, and it's a beautiful system, but yeah,
can't punch holes through floors all the time, right?
Mm-hmm. Not in clean rooms, especially.
That's it. Well, even that, I imagine there's a certain amount of downtime for surrounding areas that are impacted that might be required to get it up to speed and get it validated. Anne, thanks you for a nice presentation. She probably missed, but does the filling station also handle bags to freeze cells in bags?
No, it doesn't. It just does vials.
Well, all these questions are pointing towards another discussion point that I was thinking of. You showed some sophisticated processing equipment. A lot of the processes that come out of our laboratories, the translational projects to get into clinical production, are often a little more open in terms of the process, right? Spins and washes often take place in tubes until you get to a certain point in a product's life cycle. Did you find a tremendous challenge to bring in some of this closed processing equipment? I know certain people on the call and interacting and even following up on these questions might be interested in Miltenyi products, for example. There's a few others that you mentioned. Did you find that incorporating the closed systems into your manufacturing, your clean rooms was an interesting challenge?
No, I wouldn't say it was a challenge. I mean, it definitely reduces the risk, you know, when you have a closed system. And well, I was just going to follow up too on the bags. We do fill bags, but we fill them manually in the clean room so that they're filled. And Meltonese Prodigy, of course, uses bags, and so you sterile weld. And so, you know, you use closed systems as much as you can, even when you have open manipulations. If you can use sterile welding from going from one bag to another if you're dispensing. But I wouldn't say we've had challenges with using closed systems.
No, that's great. Sometimes it is a challenge to work backwards, right? To work with the groups that might be bringing their projects out of their laboratories and getting them into more of the closed system equipment.
Mhm.
Yeah, very good. While we continue the discussion here, I'm just going to put on our survey slide. Um, is there any major consideration that you'd like the audience to know about, especially as they approach you to discuss the possibility of working on their projects, critical areas to focus on to help speed the entire tech transfer type of thing up?
Yeah, I think that it's always good, like the old saying goes, begin with the end in mind. And I think it's good to um, have dialogues early with Health Canada. Health Canada always says this as well, like to have pre-CTA meetings so that you know which areas to focus on, even in your development process. Like maybe you have to switch to a different raw material and you want to bring that in earlier so that you can make sure it doesn't change your product when you change the media, for instance. Or moving into more automated systems may make things a lot more efficient once it gets into the GMP facility. So it might be even engaging with some of the vendors to see how their process might be able to move into an automated system or a closed system. And talking to us earlier on too, to just say, you know, take a look at our process and what do you think? Like, where do you think that we may need to shift things in our development that would bring us closer to GMP? And then also just keeping good records. I think the more records that you keep during the development process, like if you're making sure that you record lot numbers of things and expiry dates and catalog numbers and details during your process, it makes things easier to transfer to us eventually for GMP. And don't forget about the QC assays. I find a lot of times people are super focused on the process itself, but QC assays sometimes can take longer to actually get transferred and qualified and implemented, or find a contract lab that can do the QC testing for release. So it's deciding on all of your QC assays that are going to be needed and what the method will be, and then even transferring those methods to a GMP facility as well. So having good records of those assays themselves. So just some tips to keep in mind.
Absolutely. Yeah, documentation, good documentation practices, good record keeping. These are messages that we're definitely trying to echo throughout the curriculum, the training curriculum that we're building as well. So absolutely critical. Ken has another good question. The global shortages that are faced in biomanufacturing right now is definitely an issue. We hear certainly of many reports of around 24 months of a waiting period before some manufacturing projects are initiated. Have you had any specific challenges that you're facing in regards to this?
Yeah, I would say our clean rooms are currently full. So that capacity and I couldn't say that a year ago. So I think we've really ramped up. I think because we're getting busier, we're going to need more staff. It takes time to bring on those staff and to onboard them and get them trained and everything. So um, I think the pandemic has brought to light manufacturing, biomanufacturing in Canada and the world, and I think now is the time for people to pay attention and to invest in biomanufacturing and training of staff so that we can build on the capacity that's already there too. We need more capacity, but we really need to support the capacity that's all the investment's already been made. We have some great facilities. We have some great talent in Canada, and we can just build on it further. We're not starting from scratch, but we definitely have some room to grow.
Absolutely. Well, you bring up a good point, something that I know you've seen us talk about some data that we've generated within our market research in the field as well. We see such a spread, a range of quoted time to onboard newly recruited HQP. So many people report anywhere from 6 months all the way up to 16 months, with sort of the median around 8 months to onboard an HQP. Do you feel there's a way that we can alleviate some of that burden, that we can compress maybe some of the onboarding, or maybe we're just not, certainly your facility, you've excellence, you're great at training people. Maybe there's a better way that we can approach training.
Well, I think as I mentioned, there are some standard techniques that every facility is going to require for production. I'm talking about specifically. You could branch out from there for sure into QA, et cetera. But starting with just production would be like training in aseptic technique and having them actually pass that media fill. Cell counting, so hemocytometer cell counting, people learn about that. They do it in the research lab all the time. But how do you know if you're accurate or not, or reproducible? Or is it reproducible between person one and person two? And that's where some of that competency comes in. So if you have two people, a trainer and a trainee, and they both count the same cells, and you decide the cell count should agree within X percentage, and you're able to count different types of cells and different concentration of cells, etc. If people could go through a training program and have that designation that, yes, I passed this criteria, I can count cells accurately, those are all things that would really help, that then we wouldn't have to go through those types of competencies. Maybe we would have to, I'd have to talk to my QA manager first to decide if we'd have to repeat that. But I mean, it would bring us, ahead of the game if we knew when we were hiring somebody that they had those competencies. So I think that type of training, and like I said, people do cell counts all the time, but we can't just count on that they say that they can do it. We have to have proof that it's being done correctly.
That's right. And so, what I'm hearing is delivering someone who has effectively certified skills. That you
Yes.
can say we validate this training program, we've qualified it, we trust the source of the training, and the person is as job ready as possible as they get to your floor. So really not having to take steps backwards in the training life cycle and re-educate them on how to do the process according to your specifications.
Yeah. And I think if it was a consistent, like CellCAN, for instance, if you delivered that type of training and it was consistent, then if you hired someone from that program, you would know what the criteria for that program was. And
Absolutely.
you would have a consistent type of training across the country. And that's a great sell, I think, even internationally, to say when you come to have contract manufacturing done in Canada, there's a consistent bar that has been passed as far as certified staff and training.
Absolutely. And maybe it even goes back into sampling, right? And not just the actual counts themselves, but if they're responsible for producing that specimen for counting, then you need to standardize the techniques all along the path.
Yeah.
So,
And
excellent point. So I see we've come to the end of our time here. Gayle, this has been absolutely spectacular. Thank you so much. As always, you've done a fantastic job, and we really look forward to advancing the curriculum with you and bringing a lot of these points we've discussed today into fruition. Again, I would ask everyone, if you have a chance, please don't hesitate to complete the survey. This is how we build a lot of the content that we put forth and bring it out the door and to your laptops or your offices, wherever you're able to work these days. There's a QR code if you prefer to scan to get you right to the SurveyMonkey. But we really thank you for your time. On behalf of CellCAN, thank you so much, and we look forward to talking to everybody soon. Thanks, Gayle.
okay, thanks. Bye.